Now we're going to talk a little bit about microbiology, media, how specimens need to be handled, and streaking for isolation. So when we're talking about collection of specimens, we want to talk about criteria for good collection. So some of those criteria are laid out here. We want to obtain the specimen before antimicrobials, because what are antibiotics made to do? They're made to kill bacteria. So if a patient has an infection that we're wanting to culture and potentially identify a pathogenic organism, if the patient has already been started on antibiotics, then it's going to affect the results of that culture. We want to collect it from the optimal site, so from the infected site. We want to get the affected site without the surrounding contamination. So if say it's an open wound on a patient's leg, we want to put the swab in the wound, in the center of the wound, and avoid the skin area around the wound. Optimal collection time. So we want to obtain during the stage of illness that the most organism would be present. So an example of that would be when the patient's fever is up. We need sufficient quantity of specimen for the testing that needs to be performed. We want to have it transported properly and in a container that will be able to maintain viability of the specimen. So we either need to properly preserve the specimen or get it to the lab as soon as possible. The specimen ideally should be set up within 2 hours if a preservative transport media is not used for collection. So they make specific types of wound culture swabs or urine culture tubes, etc., that will have preservatives in it. So if the nurse or doctor or whomever is not able to get the sample to us quickly, at least it's in a media that will be able to keep the sample viable. Sufficient clinical information. So this is extremely important. The site and time of collection. So the site, especially, for instance, where on the patient's body did the sample come from, that's going to help us determine the type of organisms that we are looking for in the sample, in the culture, because, depending on the side of the body that it was collected from, we could be looking at normal flora that we need to distinguish from potential pathogens. And then the time of collection, that's always important, that's notated on the container, the specimen container. If a clinical specimen comes into the lab and it does not have the site listed on it, you need to call and find out. Call the nurse that collected the sample, call the doctor, whoever you need to get in touch with to find out the site, because it's also, not only is it important in determining how we determine pathogen versus normal flora, but also what plates that we set the culture up on. Has to be labeled properly, of course, so that has to have the patient information and the specimen information. So with the patient information, we want at least the patient's name and date of birth. They can also put the medical record number, who collected it, and then we want that specimen information that I just mentioned. And then it needs to be collected in sterile containers. So we don't want to contaminate the culture. One specimen type that we would not worry about that as much as with stool specimens. Okay, so some tips for specimen rejection. If the wrong preservative or collection media is used, trying to think of an example. Say a wound culture is collected and they collect it on a throat swab that doesn't have a preservative in it, that might be reason for rejection. Insufficient quantity, so if we don't have enough sample to actually perform the tests that have been requested by the doctor, and in some cases we might be able to call and request additional sample, if it's an easily obtained specimen. In some cases, if it's not an easily obtained specimen, you can call the doctor and say, hey, we've got this specimen. You've ordered all of these tests. We don't have enough specimen to perform all of those tests. Let me know which ones are most important. Let's prioritize now. Tell me what you absolutely need and what you don't, and then you just might have to go from there. If the sample's not delivered to the lab in a timely manner, so especially if it's not in some type of preservative media, the specimen may no longer be viable. If the specimen's contaminated, if the specimen's not labeled properly, so either the requisition and the specimen container don't match. So, especially if we receive specimens from outside facilities, there's a paper order form that has to come with that. So maybe the information on that order form does not match the information that's on the specimen itself. If the specimen isn't labeled, we have to reject it. So all of those are reasons for specimen rejection. Now we're going to talk about anaerobic collection just a little bit. We'll get into this much more later on. So you can read about anaerobic collection and processing in the book. We're not going to go into it too much right now. For now, mainly just know that if a specimen is delivered for anaerobic processing, that it must be in a proper anaerobic collection or storage device. So if the doctor suspects an anaerobic organism, meaning one that does not grow in the presence of oxygen, then we have to receive that specimen in a collection or storage device that does not introduce oxygen to the sample. That could kill the organism if it's an anaerobic organism. And it has to be from a site where anaerobes are not considered normal flora. As far as transporting specimens goes, like I've stated a couple times already, we must be able to keep the cultures alive until they can be tested. So ideally, specimens should arrive in the lab within 30 minutes of collection. That's not always possible, especially if it's like a home health situation or an outpatient type situation. Preferably within 2 hours. If not, then the specimen needs to be properly stored or preserved. So some ways that can happen: transport containers such as a urine cup, a sterile cup, for sputum, it's going to be the same type of cup that's used for urine. Stool container, that can be the same as well. Blood culture bottles, and then some different types of holding or transport media. So they're generally not going to encourage the growth of the bacteria, but they can at least keep them viable, keep them from dying before we can set up the culture. So Carrie Blair is one type of holding or transport media. This is for fecal specimens. It's for the recovery of GI pathogens such as Salmonella. Stewart's is another commonly used for transporting specimens suspected of having gonococci, which causes gonorrhea. Also used for transporting throat, vaginal wound, and skin swabs that might contain fastidious organisms. And fastidious organisms are just those that they're a little more finicky with their growth requirements. They may need a little additional supplement or something that helps them to grow. Amy's charcoal is modified from the Stewart's original formula, where there's charcoal in the collection device that helps eliminate the metabolic products of bacterial growth. And then you have pre-reduced media or anaerobic transport media. So these would be appropriate for your anaerobic organisms. Oxygen is removed from these collection devices so that they would be appropriate for recovering anaerobic organisms. Okay, and here's just a few pictures, a few examples of some different types of swabs that can be used. You can see in the bottom of each of these tubes here, there's a gel. So that's the preservative. The one on the far right here is charcoal. So that charcoal that's in the media, again, it just helps remove some metabolic byproducts from some of the bacteria. So just a few examples there. Okay, next, a little bit about shipping. So you can read about that in the book. We're not going to go into it a ton right now, but just know that, when we're shipping biological products, whether that be like human tissue samples or bacteria, there are a lot of rules and regulations about how that has to be done, how they have to be packaged, and how they have to be labeled, and how they have to be shipped. Okay, now we're going to get into different types of media. So this is what you guys probably think of as like your Petri dishes. This is just what we use to culture these organisms. So media is classified according to 3 properties. So it can be classified according to the physical state, the chemical composition, or functional type. So physical state, we've got a few different types. So you've got liquid or broth, where you have nutrients that are dissolved in water. So, and you would inoculate these with your swabs or your bacteria, with your specimen. And bacterial growth is indicated by change in appearance from clear to turbid. So if the liquid broth media started out clear, we inoculated it with a wound culture swab, and then it incubates overnight, we come back the next day, and it's turbid or cloudy, then that means that the bacteria has grown in that media. Solid media, so we have to use an agar base. So this is a solidifying agent derived from algae. It's flexible and moldable. You can add additional nutrients to the media or inhibiting agents. So this could help like the additional nutrients would help certain bacteria grow, whereas the inhibiting agents would help suppress particular organisms. So then you have to heat the agar. You can add your supplements, you pour them into the Petri dish, you allow it to cool, and then it becomes a solid. And that's what you end up with is your agar plate. And then semi-solid are also available, but we won't really go into it because we won't really be using it in the lab. Okay, if we're categorizing based on chemical composition, again, we'll go into this more as we're talking about specific organisms. Certain organisms, like I mentioned a few minutes ago, especially fastidious organisms that are a little bit more finicky about their growth environment, that's where you'll see the chemical composition of the media change. And then functional types, so different functional types. So what are we using the media for? So we've got non-selective or supportive media that are going to support the growth of most non-fastidious microbes. So it's considered kind of an all-purpose media. So examples of this are your blood agar plate or sheep's blood agar. So those terms, that's what that stands for, and those are kind of interchangeable. So those are the standard non-selective media used in the United States. Then we have selective media that are going to contain chemicals, dyes, or antibiotics for the purpose of inhibiting certain organisms so that the media supports the growth of one type or group of microbes but not another. So it's essentially by those chemicals, dyes, or antibiotics that are in the media, it helps the media to select, which is why it's called selective, select the organisms that will grow on the plate. So this helps with isolation and identification of specific organisms that you might be looking for. So examples of selective media are MacConkey, Hectoin, EMB, so all of those are made for gram-negative rods, and then PEA and CNA for gram-positive cocci. Okay, next we have differential media that allows grouping of microbes based on different visible characteristics demonstrated on the media. So for example, your blood agar plate and your sheep's blood agar is a non-selective, but it differentiates based on hemolysis. Your MacConkey, Hectoin, EMB, and XLD differentiate bacteria based on color changes in the media as the organisms utilize different sugars that are in the media. So all of those that I just mentioned are selective for gram-negative rods, but they are differential based on sugar utilization. XLD and Hectoin also differentiate by hydrogen sulfide production. So some organisms are able to produce hydrogen sulfide. So those XLD and Hectoin can differentiate hydrogen sulfide producers from non-producers by showing as a different color on the media. Then enriched media, this is more for finicky bacteria, so they're going to have some type of growth enhancer in the media that are added to non-selective agar to allow fastidious organisms to grow. So examples of that would be chocolate, modified Thayer Martin, which is abbreviated as MTM, and Campy plates. Enrichment broth is a liquid media that inhibits the growth of unwanted bacteria. So antibiotics are added to the media to inhibit growth of normal flora so that we can promote the growth of certain specific organisms that we're looking for. So an example of an enrichment broth would be GN broth. That's used for GI specimens, and LIMS broth, which is used to isolate group B strep from or to screen pregnant women for group B strep. Non-selective broth media is used to supplement your agar plates, or if your plating to agar media is not possible. So say you have maybe a bone fragment as your specimen that was collected during surgery. Well, you can't streak that bone fragment across the plate. So we have to use our non-selective broth media to initially incubate that fragment of bone. So we put the bone in the broth, let it incubate overnight, and then we would subculture from the broth to our agar plates. So your non-selective broth media is able to detect small numbers of most bacteria, but again, you would have to it would have to be subcultured to solid media in order to test and identify organisms. So an example of this type of media would be Thio, Thio, Thio broth. Here's just a picture of a sheep's blood agar plate. And again, sheep's blood agar plate, it's an interchangeable term. Okay, and then here's a chart for just showing some of our different examples of selective and differential media, what they select for and what they differentiate based on. So for your gram-negative bacilli, so all of these media types here are selective for gram-negative rods, which means only gram-negative rods should grow on those plates. So the plates have things in them that make sure that they are selecting for the growth of the gram-negative rod versus gram-positive cocci. But then these media are also differential for lactose and or xylose fermentation, depending on the media type, and then some of them are also differential for hydrogen sulfide production. So, for example, your MacConkey plate and the EMB. MacConkey and EMB are used interchangeably in the hospital lab, so your lab would use one or the other. There's really no reason to use both. But again, both of those are selective for gram-negative rods. So if an organism is growing on these plates, you know that they are gram-negative rods. But then beyond that, they also have lactose in the media. So they can differentiate based on lactose fermentation. So on a MacConkey plate, if the organism is a lactose fermenter, the colonies will appear dark pink. If they are a non-lactose fermenter, then the colonies will remain clear. And really, when we say clear, it just means that it does not change colors. It's going to remain the same color as the original media. MacConkey and EMB plates are both, they start out kind of a pinkish color. They do not have a hydrogen sulfide indicator in them. So the EMB and the MacConkey do not have this hydrogen sulfide. However, Hectoin and XLD, these are media that are used for recovery of Salmonella and Shigella. So again, they are selective for gram-negative rods. Hectoin plates will show lactose fermenters, and the Hectoin plate starts out kind of a green color, kind of a dark green. Lactose fermenters, meaning that the organism that has been inoculated to the plate is able to utilize the lactose in the media, and it produces a color change on the media so that the colonies turn orange. Non-lactose fermenters do not utilize the sugar, so there's no color change that occurs, so the colonies stay that clear green color. Hectoin and XLD have the hydrogen sulfide indicator in them, which shows up as a black dot. Either the whole colony will turn black, or you'll see a black dot within the colony. Salmonella is a hydrogen sulfide producer. Shigella, which is another stool pathogen, does not. So when we're looking at Hectoin and XLD plates, we're looking for non-lactose fermenters, either with or without the hydrogen sulfide production. XLD is just a little bit different. It's the same function as the Hectoin, but the sugar in the media is xylose instead of lactose. So again, same concept. So we're still trying to recover Salmonella or Shigella, both of which are non-sugar utilizers. So they're considered non-xylose fermenters. So the XLD plate starts out red. So your xylose fermenters will make a darker red. Your non-xylose fermenters, which would be your Salmonella or your Shigella, would not utilize the sugar, not change the color of the media, so it's going to stay the same original color. But again, they do have the hydrogen sulfide indicator in them. Whoops. And then again, for your gram-positive cocci, that these media are selective for gram-positive cocci, which means they're selecting organisms that are only gram-positive cocci to grow. So Columbia CNA suppresses the growth of most gram-negatives. It sharply defines hemolytic reactions. So it's kind of just an improved blood agar plate. PEA is an enriched selective media for isolation of obligate anaerobes, and then MSA or mannitol salt agar differentiates. So it's selective for gram-positive cocci, but it also differentiates pathogenic staph organisms from non-pathogenic strains of staph. And again, this happens with sugar utilization, mannitol being the sugar in this plate. So, and again, you've got that color change that occurs when the organism utilizes the sugar. Here's just a kind of a visual representation of what's happening when you've got selective and differential media. So you start with a mixed sample. So there might be multiple organisms in there. You've got your general purpose media here, and you can see all the little colonies that are growing there. So everything's growing. It's not selective for anything at this point. But on your selective media, you can see only a couple organisms are growing. So we inoculated the same sample that all of these organisms originate from, but on this media, because of different things that have been added to the media, it only allows certain organisms to grow. And then the same idea over here, where we start with a mixed sample, put it on general purpose media where everything grows, but then differential media, so we're differentiating based on color changes, and usually it's sugar utilization, so you can see the different colors of the organisms that grow there. Here's just an example of your MacConkey plate where you've got a non-lactose fermenter, so it has not utilized the sugar in the media, so it has remained the original color of the media. This kind of light pink is the color that the plate starts out as. Then you've got an organism in the center here that's been streaked that utilizes lactose, so it's turned dark pink. And then E. coli is kind of our little different guy here. He and well, I'll show you on the EMB in a minute. He just produces a different reaction. It's still a gram-negative rod. It's still a lactose fermenter, but he really changes the media around him also. Your EMB, again, like I said, your McConkey and your EMB plates are kind of interchangeable. Your lab would use one or the other, but they're going to tell you the same information. If an organism is growing on the EMB plate, that means that it is a gram-negative rod, because the media is selective for gram-negative rods, but then is also differential based on this lactose fermentation. So on your left-hand side here, and the EMB plate starts out a little bit darker pink than the McConkey does, but non-lactose fermenter here, so you can see the organism that's streaked on the plate, but it hasn't changed colors. In the middle here, we've got a lactose fermenter that has turned the media kind of a dark purple. And then again, E. coli is the only one that will do this. It'll form this metallic green sheen, but that's considered lactose fermentation also. We've got our Hectoin plate, which again is selective for gram-negative rods, but we're using it to differentiate for Salmonella or Shigella. So it's got lactose in the media. So and it starts out this green shade right here. So where it has turned orange right here, we've got growth. So we know that every organism that's growing on this plate is a gram-negative rod because the media is selective for gram-negative rods. But then lactose fermentation has occurred right here with this organism. It's turned the media orange. But on Hectoin, we're not worried about the lactose fermenters. We are worried about the non-lactose fermenters. So you can see around the edge of that black dot there, you can just barely see the kind of clearish green colony, and then it's got that black dot in the center. So that colony is suspicious for Salmonella because it is a non-lactose fermenter that produces hydrogen sulfide. And then here's just another picture of Hectoin with only Salmonella growing on the plate. So you can see the media has not turned orange, so there is not a lactose fermenter on there. And we've got those green colonies, and you can see some black there also. XLD, again, it's the same concept as the Hectoin. We're using it to isolate Salmonella or Shigella. It starts out as this red color. They've inoculated it with E. coli here, so it's not really changed color, but then we've also got Shigella and Salmonella that they've inoculated over here. So we're seeing the hydrogen sulfide production, so that's suggestive of Salmonella. Next is our chocolate agar plate. Again, this is used for kind of finicky or fastidious organisms. Here's our thio broth. So that's what it starts out looking like. Okay, and then I think the last little piece of this lecture that we're going to talk about is streaking for isolation. So we do this for the purpose of quantitation and identification. So we're from that original mixed culture. So we've set up a culture, and again, I'll just use the example of a wound culture or a wound specimen. We've inoculated our plates and we had multiple organisms growing, but there's one within all of those mixed organisms that we feel like is a potential pathogen. So we need to isolate. We need to get that organism by itself on a plate so that we can do further testing so that we can identify it. So the way you do that is from that original mixed plate, you're going to take one colony that you want to isolate, streak it onto another plate with a loop into 4 quadrants. So that's how that's done. We've got 1, 2, 3, 4 quadrants. And then when we quantitate our report, when we send out our final report, we have to quantitate how much of that organism was growing. It kind of gives the doctor an indication of how severe the infection is, but we quantitate from the original culture, not from our subculture, but from the original culture based on how many quadrants the organism grows in. So again, this is just an indication of the severity of the infection. So we quantify it as either 1+, 2+, 3+, or 4+, depending on, from that original culture, how many quadrants is that organism growing into. So again, the steps for culturing microorganisms, we have to inoculate from our original specimen onto the appropriate media. We have to incubate that culture. Then we have to isolate, and it's not always necessary that we have to isolate. Sometimes we have organisms on the original media that are isolated well enough to do our testing from, but if necessary, then we isolate, inspect and do our testing, and then identification. So that's basically what these couple slides say, so I'm going to skip over them. Then the very last thing here, hemolysis. So blood agar contains general nutrients and 5% sheep's blood. It's useful for cultivating fastidious organisms and for determining the hemolytic capabilities of an organism. So some bacteria produce exoenzymes that lyse or destroy red blood cells and degrade hemoglobin. So those exoenzymes are called hemolysins. So bacteria can produce different types of hemolysins. So a beta hemolysin breaks down the red blood cells in the media and the hemoglobin completely. So the result of that is a clear zone around the bacterial growth because we've got sheep's blood in our media. This organism produces a beta hemolysin that is capable of hemolyzing or destroying the blood cells in the media. So that leaves that clear zone. That's referred to as beta hemolysis. Alpha hemolysin only partially breaks down the red blood cells. It leaves kind of a green to brownish color behind around the colony itself. So that's referred to as alpha hemolysis. And that green to brownish color is caused by the presence of biliverdin, which is a byproduct of the breakdown of hemoglobin. And then if the organism does not produce hemolysins at all, it means it does not break down the blood cells either partially or fully. So no clearing occurs, no different color will be produced in the media. That's called gamma hemolysis. So here are a few examples. So that's beta hemolysis. You can see where the organism has been streaked here, and then around where the actual colony is, you can see on this image it kind of looks yellow, but it's cleared. So it's broken down all of the blood in the media in that area around the organism, and it's clear. So in the middle here is alpha hemolysis. So you can see the little tiny dots there are the colonies themselves, but then around the colony is that kind of brown to greenish color. Over here in C, no hemolysis has occurred, no breakdown of the red cells, so all you see is just the organism on the plate there. Okay, and then just a few little review slides here. Which type of media has things added to it so that only certain types of bacteria will grow? That's your selective media. Why do we streak for isolation? We have to do that so that we can get certain bacterial colonies by themselves so that we can perform additional testing for ID and sensitivities. And then which type of media do we determine hemolysis from? That is always and only from the blood agar plate. You may be able to tell that hemolysis is happening on other types of plates, but we only grade it from the blood agar plate. And that's the end of Unit 1, Lectures 1 and 2. So the rest of Unit 1 lecture will be on Staphylococcaceae, and that will be in an additional lecture video.